Good afternoon, everyone. I'm hoping that there will be many attendants of that very special workshop. We've already had three wonderful speakers in the morning, and here we are going to have a brilliant teacher. I remember the first time I saw one of his online courses, I thought that's our person, and we have to ask him. And fortunately, he found the time to join us today, and his name is Jason Williams. Let me introduce him a little bit to you. So Jason is Assistant Director in External Collaborations at Cold Spring Harbor Laboratories DNA Learning Center, where he works to spread hands-on biology education internationally. Jason provides training and support to scientists and educators, helping them leverage the most advanced tools and the best methods for research and education in data-intensive biology. Additionally, he serves on several committees and boards for projects that advance science and science education, including his service in the steering committee of the Software Carpentry Foundation and as an instructor of software and data carpentry. Jason is co-creator and lead instructor of the Science Institute and Yeshiva University High School for Girls. Jason, the floor is yours.
All right, thank you very much, Lucas, and everybody who is tuning in. Let me just make sure that folks can see me now. I spotlighted myself. So thanks very much, Lucas, and the organizers for inviting me to do this workshop, and for everyone that is watching online. We'll be together for the next hour. I'm going to start with some slides to give you a little bit of background on transcriptome analysis and show you the tools and walk you through a pretty well documented tutorial. So really, if you have questions, you can take them anytime, and Lucas will be feeding those to me, and I will try to respond to them as soon as I can. But I'll remind you that all of these materials are really online for you to view anytime, so you can decide. Some people might try to follow along to some extent, although you'll get bogged down when certain steps do take some time to complete, but you'll have everything so you can return to at your own time. So with that said, I am going to go ahead and share some slides for the first portion, and then I will go into sort of walking through the steps of the materials and letting you see how everything works. My email address and social media is there. You can contact me if somehow I don't get to your questions today. Okay. So there are a couple of goals for this session over the next hour. One is I'm going to introduce you to the resources, this project that I've been working on for a while called SciVerse. And these are free resources for everyone to use. So you could certainly go ahead and make application of them. I'm going to give you a broad overview of RNA-seq practices. So things that you may think about if you're already doing or will be doing some type of transcriptome analyses. And in particular, I'm going to be introducing and examining the Kallisto and Sleuth workflows. These are tools related for bulk RNA Seq. I won't necessarily be discussing a single cell today, but these actual pipelines and workflows exist for single cell as well. And it's really about getting you to identify these tools and resources so that when you are ready to work with your own data, you are positioned to do that. All right. So quickly, just a little bit of background on this project, SciVerse. So this is a project based in the United States where I am. It's about $115 million invested over a number of years. Currently, there's about 90,000 users across 2,000 institutions and more than 5 petabytes of user data that are managed on that platform. It's freely available to the community, including national, international collaboration. And we have partners in terms of their own separate setups at SciVerse UK and also SciVerse Austria. So these are different places and networks which are using the platform. Of course, you can always use SciVerse's US as well. This actually started back in 2008, where the project had a different name called iPlant. It was actually formed to serve the need of plant biologists in particular. And then as the project grew and was renewed, et cetera, took on a larger scope of really thinking about doing this for the entire scope of biological sciences. Although we will only be covering a small subset of the materials and resources available, there's an entire product stack of everything from the image analysis to various tools for learning, APIs. We're going to be working a lot today in this thing called the discovery environment, and also working with some of the data products. And if you were more of a bench biologist, you'd be working at this sort of layer of these nice, simple and easy to use web interfaces. But also for people who are more on the end of developing their own pipelines, you can actually access some of the services and foundational capabilities, everything from containers and container orchestration to working with federated storage and so on and so forth. So there are really a lot of capabilities depending on what your level of skill is. I'm assuming the majority of the audience will be, in this case, microbiologists, but for those who do have a little bit of a developer toolkit or interested in applying it in more customized ways, those are things that you can do. Okay, so like I said, we have now less than an hour. And so what can I get you in that amount of time? I really want you to be realistic. I have to be realistic so that we don't try to cover too much without really covering anything. So I'm going to talk to you some basics about the practical software and what I would say is data hygiene, just how to really think about your project before you take a deep dive into it. Or if you're already doing these things on a day-to-day basis, maybe even make you aware of things that could make that science more producible. I'm probably going to get some basic concepts across to you, foundational ones about RNA Seq and particularly these workflows. If this was a cooking class, I'd also in an hour succeed at teaching you how to make really wonderful fettuccine Alfredo. What I cannot do is really tell you everything that you need to know about RNA Seq. So please keep in mind that this presentation is limited. I'm not going to be covering every single nuance in detail, but just really let me know. There was a question about the presentation slides, and so we can find a way to make those available. I guess maybe they'll be either posted to the website or you can email me, williams@cshl.edu, but I'm being told that they have a way to post them, so we'll make sure that you come back. I also would not try to do sword juggling in an hour. It would come out pretty messy. All right, so what are the compromises we had to make in order to get this to work in the 1 hour? One, we're focusing in this case on a eukaryotic organism, and since this is a microbiology conference, I chose Plasmodium to look at a nice single cell creature. And we're going to be using software, this Kallisto and Sleuth, and it's really a very simple and straightforward pipeline, which is excellent. And it manages to do that without compromising its accuracy. So that's great. We're also doing everything really on the Cyverse platform. So there's no tools to install. Everything is running on the cloud. And I think, and I hope that as biologists, we're getting more comfortable with that, because often we're working with large data sets that we cannot manage on a computer. In terms of prior knowledge, as we get towards the end, when we do the visualization and play around with some of the data, being familiar with R will be helpful there in RStudio. But if you don't know those things, it's not that you won't be able to follow along, but you probably will need to get a little bit more familiar before you make lots of changes on your own. Okay, so let's start with these two things, Bulk Rna Seq, and we've got 4 points to cover there, and computation, how to really get this started on your own. We're going to go through this step by step. So, introduction to the Bulk Rna Seq. There are 2 papers that I actually recommend, so I'm keeping this up for just a second to think about this in the background. There's an older one, the 2016 one from Kness et al., a survey of best practices for RNA Seq. Of course, the technology has changed. Of course, things have been updated, but this still is pretty much a really nice survey, all in one place of practices that are really still what you need to be doing. Again, if you were using single cell, there's a new set of practices that you might want to also think about, but still, I think a lot of the fundamentals are there. And then this more updated one, 2019 from Stark et al. If I could only choose 2 papers to sort of tell you, read these as homework, these would be really good ones to take a look at. So they're up there and you will be able to find the slides later. the first thing that I think Rna et al. States that I wanted to emphasize here is that there is no single analysis pipeline that can be used in all cases. So that's the, a key thing that's right there in the abstract. It's really, I think, important to understand the fundamentals of how any software analysis works, because either you're doing it yourself, and if you're a novice and it's your first time, then you don't know what you don't know. And so you can get lost and maybe make some bad assumptions. But even if you're working with an expert, so if you're collaborating with somebody who does have expertise, they may not know your organism or your system as well as you do. Just to sort of say, for example, if I like to go back and forth between my sides and this for emphasis. So even if you you come up at the end of your analysis and you have a list of genes, and some of them don't quite make the significance cutoff, right? A collaborator or somebody else, might just throw them away and not consider them. But you, as a person who knows your system and knows the background of your system, those may be genes that are actually really important and they're close to significance, but not quite. It's not exactly cheating to say, well, maybe I need to design an experiment with more power so that I could test if these really are significantly differentially expressed. So just keep in mind, there's not one single right way to do that, although for the sake of my presentation, we settle on one way. So just keep that in mind. Okay, back to sharing. The other thing about this experimental design is that, all of these things really became possible starting in around 2008, which is actually, incidentally, where the first sort of paper, I think you could call an RNA-seq paper, really came out, because we can actually sequence all of the things that we want to do at relative cost, right? We know that the cost per megabase has come down quite a bit, probably can go lower, but maybe the sequencing companies don't want it to go too low. But it's really relatively cheap to generate some of these data sets. This figure, also from Rna et al., sort of breaks things down. This sort of top color talking about before you even design, or really as you're thinking about designing your experiment, there are all sorts of different things, questions that you may want to ask yourself. What type of library are you going to prepare? Single end sequencing or paired end? How long of a read do you actually want to generate? How are you going to control for batch effects? There's a whole bunch of questions that you do even before you have now done the actual analysis. And then once you do the analysis, how are you going to do the quantification? What types of normalizations do you want to do? And then once you go beyond, once you end up with those analyses, how are you going to either visualize that and or take those analyses and feed them forward into something like a gene enrichment? So there's a big landscape of things that you might want to think about. The first one is this one that I sort of, I guess, put in red, is that you want to make sure that the experiment that you design for your organism and for your question is actually has a potential to work. When you are doing an RNA-seq experiment, you are looking for differences in transcript level, which we assume corresponds to some level of gene activity or protein being created, if it's a protein coding gene or regulation or et cetera. What you may or may not know is how different between, let's say, two different conditions, how different those two conditions would be under that experimental treatment. The long story short is that if the difference is going to be small but meaningful, you're going to have to look a lot harder to find that difference. If the difference is going to be really, really large, the amount of sequencing that you may need to do may not be as much. But what you really want to think about is, how do I actually design the experiment so it has potential to answer questions of interest? This is an old table. It kind of depends more on your methods, but really what it's trying to get across is roughly true, that the effect size, whatever, let's say, the fold change in expression, if we're assuming, which you can't always assume, that RNA-seq is measuring expression, the larger the difference, that may affect the sequencing depths and the number of replications that you have. And it's interesting, we'll see that when we look at the example data set, which I kind of told, picked randomly, that these things may matter. And you don't necessarily have to guess, but what you do want to do to help you answer this question, one is, do you have a statistician colleague or do you have the background in stats? So if you do have particularly a biostatistician colleague who's familiar with transcriptome analyses, you can get yourself a sense of what is the sequencing depth or number of replicates I might need if I'm anticipating that the effect is going to be a small effect. If you know that the fold change of this particular gene or set of genes is within a certain range, a very tightly controlled, highly regulated, molecule, it may not change very much under your experimental condition. Other things may change quite wide range. If you don't know that or don't have any starting point in terms of no one's done this experiment or you don't know very much about the expression range, which is unfortunately going to be the case in a lot of circumstances, right? Human is very well studied. Coming from plants, I kind of know that you're envious that other organisms or model organisms may have a larger set of resources. However, one thing, and this is kind of where I started with this particular experiment, is that there are almost always some data about your organism of interest on something like the sequence read archive or the corresponding, if we're talking about folks in Europe, the European Nucleotide Archive. There may be other experiments where you can reuse data to kind of look and measure gene expression, even if those data were not necessarily created to examine your experiment. If there are transcriptome available from your organism of interest, those may be things that you can play around with, just as I'm doing today, and take a look at those genes and see what the expression values or expression range might be. Okay, other things, I'm gonna try to get through the slides. Give me another 6 or 7 minutes. Other things before we go into actually kind of doing stuff that you will think about is really your RNA extraction and how you prepare your library. All of those things matter. They're fairly well written, so I'm not going to spend time on this. I would say probably for microbes, particularly prokaryotes, there probably are some differences that you need to pay attention to, because a lot of these protocols are things you want to make sure that you're using something that is specific if you're using a prokaryote. How things like polyadenylation work, or how ribosomal RNAs might be extracted from a bulk collection of like a culture may be different for you. So make sure that you are using something very specific. The next thing is that the sequencing is going to happen. I think that's fairly straightforward to nowadays with things like Illumina. In the case of which you're using long read, you may be using, PacBio, you may be using now Nanopore, which opens up a whole other range of how you might do sort of this digital gene expression analysis. But these are all things that you may want to think about, particularly things like standards. That may apply in the sense of, can you use a spike-in, which is essentially a known amount of an RNA that is not necessarily something that you care about from maybe a different organism that something's not going to contaminate, but can you, if you have a known truth of what different RNA or cDNA concentrations are you really confident that your experimental setup and sequencing setup is measuring those things accurately? So those are good things to pay attention to and give you some controls. So I just mentioned there that usually in most cases for this type of procedure, getting rid of ribosomal RNAs or things that you don't care about are really important. Read length and strandedness. A lot of times being strand specific, we're caring, if you think about like, not necessarily microbiological systems, but sometimes people think about things like phasing and where you're actually dealing with animals or plants. So some of these may not be as important. The paired end sequencing may be good, especially if you're thinking about you don't have much genomic information, but if you're working with something well annotated, single N may be pretty fine. Batch effects, again, making sure that the samples are pooled and processed in parallel in a way where you don't throw off your transcriptome analysis because it's actually one flow cell was washed a little bit differently one day than the other, and you throw things off. And I mentioned we'll see that. Okay, almost done with the slides, or at least these portions of them. So there are, two major approaches to doing the analysis, because at the end of the day, you're trying to generate a count of the number of RNA species and then figure out how they align to the known reference transcripts and so on and so forth. The traditional, or the way that came actually out of people's familiarity with designing software for genome assembly was this idea of doing a whole alignment of your individual reads to the entire genome. There was the top hat and cufflinks workflow, which there is an updated version, which is fine to use. There are other methods now, and beyond top hat 2, like star aligner and things like that. These tend to be a little bit slow and computationally expensive. And they're also, and oftentimes guided by reference genomes or reference annotations. So you may need those reference genomes or annotations, and you may not always have that, especially working in a sort of non-model context. So the one that we're using today, this Kallisto and Sleuth, this came out in 2016. It's still used and updated, which I think is really important to use software that's actively maintained by the community, so you can get help and that somebody cares about your results. And this one was by Rna et al. In 2016. And I will not go deeply into all of the mechanisms of it, but essentially this idea is something called, well, I mentioned there's pseudo alignment. And it's the idea that given a transcript here in black, we actually do an alignment to the entire transcriptome. So not the genome, but the entire set of transcripts that we provide to do an analysis. And then in this case, the 3 different colors, I hope they're colorblind friendly, this sort of pinkish, bluish, and greenish. Imagine that your transcriptome had, these were the different transcripts, and the read in here in black, you're trying to align it. Well, what it does is it asks the question, which of these transcripts could have generated a particular read? And it breaks the transcripts down into kmers, so short segments, often about 31 base pairs is I think the default, what we'll be using today. And it sort of scans along the reads and the set of transcripts. And if a transcript could not have generated this read in black, then it basically says, why bother to count? Why bother to do the alignment to the rest of the transcript? If 60 base pairs in, I realize that this transcript could not have generated this read, I'll stop looking at it and I'll look for another transcript that could have. What this ends up doing is it's an enormous time saver, and it actually increases the accuracy, and it's actually very robust to sequencing error, because any given error that you might have in a read, it's unlikely that it will cause you to mess up this process and erroneously discard it. The analogy, and also, let me just say before I say my analogy, it also turns out that you can tell quite early on, sometimes the first 1 or 2 kmers, before you can discard most of the reads as not possibly having generated this read. So it's the very beginning and very end of this matching process where you can tell what you're looking for. In my mind, it reminds me of this sort of a little mental trick. If you're a native English speaker, you have no trouble reading this paragraph where all of the words are sort of jumbled except for the very first word, first letter and the last letter. Your brain basically fills in the rest. I couldn't believe that I could actually understand what I was reading, so on and so forth. It turns out that very little information is actually needed for you to make a decision, and this is what happens. This is why you get the tremendous speed up in doing pseudo alignment versus full alignment. And so, whereas the older methods took on the order of hours to do a really large analysis, basically you can in a few minutes be working through tens of millions of reads. This is really nice because for the few parameters that you might need to change with Kallisto to see if you can increase sensitivity, there's not a whole bunch of bells and whistles and knobs to tune on this one. You can spend one day going through a couple of different sets of parameters, seeing if you get the results, well, not that you're cherry picking, but seeing if you're getting results that look good, and then going on from there. So this is basically showing you that depending on the variance of the sample, you just get this linear correlation that we really are able, along the breadth of the possible variance that is there between different subsamples of the data, you really get this nice linear path that shows that you're picking up in terms of sensitivity what you hope to pick up. The other half of this workflow, so the Kallisto workflows you'll see in a bit, just deals with counting the reads and doing the quantification. The Sleuth workflow then takes all of your individual experiments, so all of your different replicates and conditions, and then develops a linear model where the idea of this linear modeling and visualization that we'll do in the second half in R and Sleuth, that allows you to separate out variance that's due to technical variance and really isolate, hopefully, what is the biological reason for the difference of two samples or however many samples you have, and finding that. And again, in some ways, maybe this should have come out to, this should have been an earlier slide. It's a matter of looking through any given set of transcripts, seeing what reads match, and coming up with these, what they call equivalence classes. Go read the paper if you want to get more into that, or I could take questions as they come, but that's sort of the technical meat behind that paper. Okay, so here is how this works to get to what will you need to do. There's three major steps. I'm going to go through everything, but it's something that one can get their head around. The very first step is going to be getting your data onto Cyverse. I can demonstrate that in a little bit, and it's not all that difficult. Even if you have maybe gigs and gigs of data, you pay the sort of price time-wise to upload that once. But once you have that data set on Cyverse, it could be as easy as drag and drop with something like Cyberduck. The second thing that you're going to do is there's really only a couple applications, one if you're lucky, really two if you're going to use Sleuth, but that you're going to use in the discovery environment in order to, this is the Cyverse platform, in order to do the analysis. Now, you could use these tools. These tools are built at the command line, but the discovery environment is a point-and-click interface. So even if you don't know command line, you can go ahead and use these tools. And then the very final thing is we're going to go ahead and then analyze the data and visualize it in Sleuth. And I mentioned today the experimental data. I just found the submission on the Sequence Read archive. So just so you have in the back of your head. So this is Plasmodium falciparum, which you know better than I as microbiologist, is the organism behind malaria. And I don't know if all the results were published. This was submitted in September, it looks like, and I didn't see the corresponding paper yet, so it's like registered. So we'll see. It's nice data that I was able to use. So that's what I have imported into the discovery environment. Keep in mind, like I said, that we're going to be looking at genome. We're going to be looking at pseudo alignment, so we only need a transcriptome, not a genome. We're going to visualize things. I'll come back to that later if I have to, but now is really the time that I am going to go ahead and take you through this. So let me point you to where all of these learning materials are, so that as you try to do this on your own, after presumably this workshop, you know where to go. So the best place to go is to the Cyverse Learning Center. So actually, if you go to the Cyverse website, you can get there, but it's just learning.cyverse.org. But from the Cyverse website, maybe you've already account, and if you go to this Learn thing and learning materials, you will find our website. And under tutorials, there's a nice one called Rna Seq with Kallisto and Sleuth. And it's a general purpose tutorial that will take you through all of these different things. Now, the data set in this particular tutorial is a different data set, but the instructions are largely the same. The first thing that it's going to tell you is that you need to import some data. So let me sort of take you through what you need. Let's go to home first. There we go. Okay, so kind of let me sort of take you through what you need. You are going to need to import data into the Cyberse data store. Now, actually, if I show you where those learning materials are, they're linked here, but I'll just pull it up. In the platforms here for the data store, there's a whole walkthrough of how to upload data. You could use something here called CyberDuck, and it would take you through configuring that. So what does this look like on my desktop, which I'm bold enough to kind of share for a moment? I have this CyberDuck connection. This is just a window. I'm sorry, I can't make it bigger. Open, and what I'm actually viewing are all the data sets on my Cyberse data store. On the Cyberse data store, you can have for free up to 100 gigs of data storage. If you need more, up to 10 terabytes, you just need to fill out a form to give us some justification. But once you have, like I just happen to have these files on my desktop, once you have your reads or your files of whatever size, it really is as simple as I'm connected to my data store here, and I can drag and drop these, and it will go ahead and start an upload or a download. So you're going to spend 20 minutes, an hour, however, whatever the size of data set that you have, importing this data into the Cyberse data store. Let me go back to, if I find it, go back to the lesson here. So when you upload to the Cyberse data store, you might want to see that data. And so what we're going to do, again, let's show you from the Cyberse homepage, is we're going to go to the Cyberse discovery environment, which you might have looked at if you signed up for an account. But we're just going to go ahead and click here. So I'm going to say launch. Okay. So this is the Cyberse discovery environment. And on the left-hand side here, there is sort of a menu that you can go back and forth. And if I click on data, what I'm going to see in that menu is the same thing I saw a few moments ago on my CyberDuck interface. And this data set, if anybody's really interested in it, I can share it with you. But let me go make this a little bit larger. But the data set that is in the tutorial is already public and freely available for use. But I put my data set in this SFAM demo. Okay. And what I needed to do first is I needed to import data from the sequence read archive. So these are the actual data sets that I was able to import. And these are just FASTQ files. So however you get your FASTQ files, let's assume that you have them. So what's next? Okay, let's go to the lesson and see what it tells us. Let me close some of these things I don't need. Okay, so I'm going to go to the next step. On the lesson, well, let's go and see it. All right, so the very first thing you need to do, once you have your raw sequence data, your FASTQ files, there is an optional set that's not mentioned here, but you may want to actually do some quality control of your data. And there it mentions that you can use an application called FastQC. I'm going to show you how that works. It's optional in the sense that if you've already done this quality control, and often your sequencing company will give you results so you know how well the sequencing worked. But let's say you didn't know, and in my case, since I was importing data that I have no idea how good it was, I needed to do that. So let me show you how that works in the discovery environment. So in the discovery environment, If you want to use an application, you could search for it in this box. So if I type in FastQC, which some of you might have used, you could see that I might find some different options or variations of this application. And let's say I wanted to look at all, in my case, I have 6 FastQ data sets. I could click on those. And the way that analysis works in discovery environment is sort of a step-by-step process. So first, it gives it a name to my analysis. And I could say, you know, this is the SFAM demo. I could say, you know, this was for live demo. And I encourage you, especially since, you know, we're not doing things at the command line, and that makes it inherently hard to document things, you know, add comments, make descriptive titles. Then it tells us that it's going to output this data to a place called analyses. This is the default, but you could browse, and let's say that we have created, which I did, I created this Plasmodium demo folder, so I could say, make sure those results come out there, but I'm not going to change that right now. I'm just going to leave it as the default. I'll go ahead and click next. This one doesn't have very many parameters. It only really needs to know where are your FASTQ files. So I'm going to go ahead and browse. It automatically opens to the last place that I was at, so that's convenient. And I could say, well, let's just look at the raw things. And I'm just going to choose one for the sake of time. And I'll say, okay, that's the file that I want to do an analysis of. And I could say next. Now, there are some options here, which you don't have to change. But if you have an idea that, ooh, okay, this is a parallel program. If I have more cores, it's going to work a little bit faster. You know, that's not really the case so much with FastQC. Same thing for memory and stuff like that. There are some generous defaults here. If you need something much more powerful, you can do it. You need to ask. So I don't want to say that you're limited. So I click next, and everything looks good. I'm just analyzing this one file. I'll click launch analysis. Then what will happen is it will take me to this next menu down here where I'm actually analyzing data. So it will give me a readout of what are the different jobs that I've done in the past, what applications I've used, and what their status is. So right now that status is submitted. In a few seconds, it'll change to running. But let me go ahead for the sake of time and show you what does this look like and what should you be looking for in brief before I actually do the RNA seq part. So if I go back to my data and go back to that SFAM folder, I need to put this back. I need to keep the larger window size here. All right, so what you will end up with are some results, and you'll end up with an HTML report for each one of these. And if I click on one of them, it's gonna, since that happens to be an HTML file, it's gonna open. I don't have all that much time to go through what a FastQC report is, although if there's questions at the end, I'm informed I can go a little over and you can send us questions. But essentially what we're looking for is that most of our reads are up here in the green in terms of the quality score. So I had no idea what this data were when I imported it from the sequence read archive. So I was happy to see that most of the data are really pretty high quality. Now, as it says back in the lesson, you know, you could do additional trimming and filtering with another application like Trimmomatic, which is in here, but the data are good enough that I really don't have to do that. So let's assume that you have quality, a good set of FASTQ files. What's the next step that you need to do? So the next thing that you're going to need to do is you actually need a transcriptome for your organism of interest. So depending on what your organism is, in this case, it talks about using ensemble plants, but in our case, we're going to probably be using the ensemble protists. And you know, for whatever your organism is, you can go and choose the appropriate database. The transcriptome could be, you know, hosted by a consortium. It could be on a site like Ensemble or NCBI, whatever it is. When you find that, you're probably going to say, okay, let me find a FASTA file. In my case now, it wants these things to open up in a tool like CyberDuck. So when I go and it opens these things up in CyberDuck, which because it's an FTP site, I would go into my cDNAs that it provides, and eventually I'd find that there is a Plasmodium, you know, cDNA. What you would do if you had the URL for that is it tells you that you can go and import this into the discovery environment. So back there, if I was in my data window and I had a particular folder like that SFAM folder that I was supposed to use, wherever it is, I don't know if it loads this way. Whatever folder it is, I could go to the upload thing here and I could import that from the URL. You'll notice that there's ways to import data directly, but we do recommend CyberDuck for really large files. It's just more efficient and more robust. So for small things, this is fine, but for large things, I'd encourage CyberDuck. Okay, and what you'd end up with, Transcriptome, which is just a FASTA file. I guess since that one looks like it's unzipped, if I click on it, maybe it would show me, yeah, okay. So what you're ending up with is a really one FASTA entry with a description of the gene and whatever the sequence is. So keep in mind, I mean, if that hasn't already occurred to you, that because we're doing a alignment or the pseudo alignment to a transcriptome, your RNA seq analysis will only be good as your transcriptome, right? So if your transcriptome is incomplete, there may be genes that you will not detect in terms of expression. Okay, so what does the tutorial say I do next, right? I import that data, and then what's the next step? Well, the next step is really nice because we're going to build a transcriptome index. As it turns out, what you need to do, and you only need to do it once, is you need to basically build a lookup table so that Kallisto can efficiently scan that entire transcriptome. Now, the nice thing about this is that we can use this application, this Kallisto version 4.3.1. And actually, if we click on this, it'll go ahead and directly open that application for us in the discovery environment. And it's the same sort of look and feel. I can say, you know, this is the SFAM one, and, you know, this is for a live demo. I might change this output folder to something else, but I'll keep it the same. Goes through the license. And then what I need to do is, it's just this page that I need to fill out. So it asks me, where is the transcriptome, the FASTA file? And it's going to automatically build an index. So I just need to tell it where that is. So I'd say, okay, here's my transcriptome. It's in there. The next thing is I need to tell it what is the library. And in this case, for these data, these are single read data. Then I'd have to tell it where are my FASTA files. So I only have, I'm not going to use the second box for read to, so I can browse. And here are my FASTA files. So I can select those. It doesn't matter if they are compressed or not. These happen to be compressed. And the next thing are a number of, but not that many, different parameters. And these are described as you read through here. This tells you for a different data set, but it goes through what parameters to use and also points you to the paper for specific recommendations. So I'm managing my windows. I've got so many things open. I've lost myself. Okay. The first thing here is this thing called Bootstrap. And Bootstrap is really the number of subsamples of the data. In the tutorial, I select a low number like 25 because I wanted it to go fast, but you might choose a higher number like 100. Now, as it turns out, if you are using single N reads, you need to provide the average fragment length. This is the length that was used in library construction and the standard deviation. Now, I did not make the libraries, and these are not things that are typically reported on for data on the sequence read archive. So I read this somewhere, and it's a good enough starting place as any that you can typically use a value like 200 for the fragment length and about 20 for the standard deviation. If you want, there's something called a pseudo BAM file, and this BAM file would be useful if you want to actually visualize those alignments in something called something like IGV or IGB, any of those genome viewers. So you could do that. That will take longer to process. And if you had strandedness, you could select those. But if everything is good, then you select next, which I'm not going to do. You could select the resources, which I'm not going to do. And then you would say launch. This will take on the order, depending on your data set, could take 20 minutes, could take an hour. Most of that time is not even analysis. Most of that time is moving the data around to wherever it needs to be. But when that analysis is running and or done, you will get in this menu, and remember, the titles are obscured here, so if you don't see them, this is the analysis menu. You would get, sort of a history. So if I look here, here was the last time I did a Kallisto analysis. And if I click on this folder icon, it's going to take me to the output folder. And it's worth us taking a look at what these are. So it comes out with this index file. That's pretty big. It's 200 megabytes. And this index file you can use any time in the future. You don't need to recreate that unless you have a new transcriptome. And then for each of my sets of reads, I've moved these. So let me go actually to where they are. They are in my data window. And I should just use the search button, but I'm lazy. Okay, so what you come up with is you come up with one folder for each one of your reads, and something was telling me it wants to update. I don't need to do that right now. Sorry, Docker. What you come up with, doesn't want to, there we go. It's like, doesn't want to get out of the way. Are these important files, and it tells you on the lesson here what each one of these are. So the first one you can't really open, it's not that human readable, but the one that you can look at is this thing called abundances. And so these are the estimates of abundances, and if I go back and take a look at one of them on the TSV, we'll get a preview of it. What you have is every single transcript, right? And these are the Plasmodium gene names. You have an idea of the length of the gene, and there's a normalization that goes in because essentially really large genes will get more counts just because they're really large, right? Whereas small genes, they may have fewer reads that map, but they have to be normalized. So that normalization happens, and you eventually get a number of counts, how many reads were mapped, in a normalization number called transcripts per million. So that's one of the pretty standard ways to do the normalization. I will not go into the math here, but that's what it's doing. So what you have at this moment is great, but it's not everything you need. At this moment, what you have are those counts for every single one of the files of those reads here. What you need to do next is you need to actually do the visualization and actually the math to do the comparative analysis. So for that, you're going to be using a different application called Sleuth. And Sleuth is a command line application that runs in R. So you would click this button, and when I click it, it's going to tell me something like, I'm not allowed to run it because I already have some running. So you would go through that step that I showed you of saying, selecting your file, clicking next, and clicking launch. When I go to my analyses folder, I launched one this morning, if I click on the right button. And what it will have is this application, the RStudio Sleuth, running. So it tells me it's running. And it'll have a little icon there that says go to the analysis. So I'm going to go ahead and click on that, and it's going to go ahead and start up an RStudio session where everything should be there. Now, I have the boldness here of believing that I am going to be good enough to do this sort of live with you and not make too many mistakes and get through this in the next 10 or 15 minutes or so. So let's see if I could do that. So the first thing is that if you look in this RStudio environment, familiar if you have R, I've got my folder on the right-hand side called Kallisto results. And the other thing that was imported, actually, I should have done it differently. Is this correct? Oh, okay, I did it correct. There's something here called Plasmodium experimental design. Good. I was afraid for a second I didn't have that. So what you need to do is you need to, using, let's say, something Excel or whatever you want to use, you need to create a very small file in which you have the name of your samples in one column. So the first column must be called sample. And then you have the name of your files, which mine are just the names of the sequence data from the SRA. And then in any other column, your conditions. So you could do multivariate analysis where you have different things. In my case, what they had was wild type, and then they had a knockout where they were knocking out some calcium-dependent kinases, I believe. So these CDP, these kinases were knocked out. And if you go back and read the example data set, they tell you that. Okay, so how do we do this? So on the right-hand side, there is a tutorial notebook. And what you'll notice is that this tutorial is written for Arabidopsis. Half lazy that I didn't create a whole new notebook for you, but also I just wanted to show you, you may not be working with Plasmodium, so you're gonna need to know how to edit it anyway. So why not start from the beginning? So what you do, and like I said, you don't need to know R, but it's nice because you can kind of get through this without knowing R so much, is every piece of code that you kind of need to run is in a little chunk, it's called. You could press the play button and it will just sort of run for you. So we're using version 0.0 of Sleuth. We need to load some additional libraries for graphing. And we also need to load a biomart library because when we look at our analysis, we want to have those gene names with some additional information. Okay, so my sample IDs, I need to actually load them, and they are not. So I'm going to get rid of this between the quotes, and I'm just going to say Kallisto and hit tab, and that's the folder I need to get them from. So I need to make those corrections. Kallisto results, okay. And I'm going to run this chunk, and it's going to make my sample IDs, and those look correct, right? Those are my sample IDs. So every folder corresponds to what my sample ID is. And when I run this again, I'll see it in a moment. It's going to tell me we're going to make a table. I also need my experimental design, and this is not the right name here. It is the one I just made called the Plasmodium. And I can go ahead and run that chunk. Okay. And when I look at this, let's see, it says we're going to add the directories. I run these next 3 chunks. Here's what I'm trying to create. I'm creating yet another table. It's not the most fantastic thing in the world, where in column 1, I have my sample IDs. In column 2, I have whether the condition was wild type or the knockout. And then really what I really needed was that the path to my results are given to the computer, so it knows what to look for. I'm going to save a copy of this while I'm working. Okay, so the next thing is I want to get some other data from Ensemble because my gene names aren't that informative to me, especially I don't know anything about Plasmodium and how they name their genes. So I can use that biomart R package to bring over some of that data. So here I'm going to tell it to list the number of marts, and it tells me I've got mouse and human, and these are things I don't care about. Now in this case, it says, okay, well, that was for plants. But actually, what I really want to use is protists. So I'm going to change this once I find the window I'm in, okay, and make that protists.ensemble.org and save that as the mart. And now I see that there is something called protist mart that I could choose from, and that's what I want. So I'm going to say, change this again to protist.ensemble and then use protist mart. So it was worth, I hope, if I can do this correctly and change this. I'm going to change this to protist mart, this one to protist mart, just so that you know how to do things. And then it tells me that these are all of the different choices I have in there. And in fact, if I go a little bit further, oops, let's go back. Oh, going the wrong way. These are the list of different types of genes and things like that that I could take a look at. So I'm going to change this. I want to use the protist mart again. And then the data, the thing again is protist.ensemble. All right, and now what I'm looking for is the P falciparum. So that's really what I want, right? Okay, so I'm going to put all of those different things there. Oops, I want to change this to ProtoSmart again. I'm just keeping my variable names the way that I want them. Where did I have that? ProtoSmart. Okay. Making all those changes. So that's going to be ProtoSmart. Got to change it there. Now, when I say attributes, what I can get is maybe I want the ensemble gene IDs or the chromosome names, a description of what the gene is. Those would be really nice to see. But there are all sorts of different things I can get, maybe some Go IDs, etc. So I'm going to go ahead back here one last time, and I'm going to pull in, give me all the ensemble IDs, gene IDs, descriptions, gene names, and run that chunk. We have done no jet differential analysis right now. We're just running it. And then finally, we're putting this into a column. So what it's doing in this step at 120 is, remember a moment ago, I showed you the list of counts for those CDX, whatever the falciparum gene names are. And it looked up all of those gene names in my transcripts and then went to Ensemble and found the corresponding gene name plus any other of those information, those attributes that Ensemble has. So that way I could have a better annotation than what I ended up with. Okay, let's go back to sharing. Okay. All right. Now what I need to do is I need to I think here we call this wild type with a capital W, probably won't matter. We're just going to prepare the data for Sleuth. So we're just telling it to treat that as categorical data. And then what it's going to do, this will take a few seconds, is it's going to bring over and start to create a linear model for the different sets of count data with respect to what genotype they were, whether they were the wild type or whether they were the knockout, right? So that's what we want to do, is making comparison between wild type and knockout. That'll take a moment. I see I'm definitely going to go over, but I'm going to try to keep that to, not too long. It's going to tell you how many things, passed the filter, if anything was dropped, etc. And what we can already see here, which is a lesson in data visualization, is we have a problem. So this is a Pca plot. And what we have here, these different colors, are the different samples. So the wild type samples are this sort of reddish color, and the mutant samples are in some kind of blue. And what I expect to see is that the samples on the Pca plot should cluster together, that the samples that are actually wild type belong together, and the samples that are actually knockout should cluster together. And what I'm not seeing that. This is warning me that this sample here and this sample here are outliers. I don't know. I don't want to say this is a data mix-up, but something could have happened with those. Maybe they were both run on the same flow cell and they're just not behaving, so they're not clustering. What that's a warning to you is that there may be some sort of variance that's not biological, that's happening in these, that's not happening in this, and that may overwhelm our results. Already we only have 3 replicates, which is the bare minimum. So it'll tell me that something is fishy, and we'll explore this and then show you, we can't solve it, but we can correct for it. I'm gonna move this. There's some other things here that are plant specific. What I actually need to do is come down to here. We're gonna go ahead and create that linear model. So let me just run it through this, and that'll take a moment. And then what it's going to do now is give me the fold changes for any given transcript. And then given those fold changes, give me an idea of the significance of whether that was upregulated or downregulated, etc. Okay, it took me through. There were 2 genes that for some reason had NA values. we can look at the full results. So this is starting to tell you, all right, here's a target ID, here are those annotations from Ensemble we brought over. Here is, you know, the description of the gene, which we never had before. So that was what all that biomarked for. And we start to see these p-values and q-values, right? This one, whatever it is on the edge of significance. We can add an additional test to this. But the cool thing is that we can explore this in a Shiny application, which is where this notebook ends. And when we click on that, let me go past the browser. We have this application, and if I go here, for example, into the test table, I have a listing here of these different genes. B is the fold change, so this one is relative, you know, 1.7-fold less. And skip this update. Thank you, Docker. Let's see. I also have, you know, some ideas of variance. And Q is the corrected p-value in terms of the, so B is the fold change, and then Q is the corrected value. Now, if I can also go, I think if I put in specific target IDs, I'm just going to choose this one, and I say I want to do, I think, transcript view. I can, for an individual gene basis, I can generate a plot where I look at the expression values. Now, there's not a lot of variance. I can definitely see that the wild type and the knockout, there's differences here. If my bootstrapping was here and a little better, I might have an idea of variance. I could also go through and get a volcano plot where I should be able to see, you know, the significant genes. Now, if you look, I'm actually not getting things that are looking really, really nice here when I look at my corrected volcano plot. I have different values of expression, but everything is clustered here in this sort of non-significance. If I scroll down, I could see what these or any one given gene is. And I suspect that's because we had this outlier. So let me try to correct that live and see if I don't totally embarrass myself with my R skills. So I should write it down on a Post-it note, but it was this 668 and this other one I can't see. I think that's the 71. So what I can do back here is on this table, this sample to condition table, if I tell it to get rid of the 6.8 and the 7.1. So the way that I can do that is down here. This might be at the bottom of the screen. So I can't make that go any better for you, except to make this not full screen, perhaps. All right, what I'm going to do is I'm going to say, give me sample to condition, and then give me just columns. All right, 1, 2. I'm going to skip 3 because that was the outlier, and then give me 4 and 5. And let me just make sure that looks correct. Now, already I have, you know, so that should be correct. I'm going to save that as the samples to condition. Okay, so if I go back, and I think I have time, just do this really quickly, and rerun the parts of the notebook that generate those things. So that is, I have to rebuild this sleuth object. That'll take a moment. I don't think I needed to run anything else. And then we should be able to rerun the Pca plot. And now they cluster on the same side, right? I mean, they don't have to be in the same places, but we've got something that looks a little bit better. And then we need to go through and redo the model. Now, the problem is that we've gotten rid of a significant amount of data. If I told you that 3 replicates are the minimum, you know, we're kind of in a weird space where we don't have the best sort of thing. However, let's go ahead and do that. All right. And I'm not gonna, the results, I don't have to pull that out. I could redo the test and I could redo the Shiny app. And now, for example, when I go back, and there are other things up here, but if I go and reload that volcano plot, we're getting better results because we're able to separate out, these are the ones that are downregulated. And we see some of these, calcium kinases showing up even in the first sort of list of genes, right? So you could go through and there's more sorts of things that are in here that I won't have time to show you, but it kind of shows you the power of the data visualization and to really explore those data that you have in order to, kind of look into this experimental result. So I'm really going to wrap up. Like I said, making sure you visualize those data. I always love this little animation because all of these little graphics have the same mean and standard deviation correlation. So you can make the data do whatever you want if you cherry pick it. So you really have to go in and look to see those patterns. But I wanted to close up by saying that really everything is documented on the Learning Center there, and also to thank the respective teams that are sort of behind this at University of Arizona TAC and Cold Spring Harbor. So with that said, I think I will stay on for another minute or so. I do see, I see a question, and like now's the time to give me questions. I'll stay on for another minute or so. One question is, do you have any recent publications that apply Kallisto and Sleuth workflow? I mean, there are so many, literally just Googling RNA Seq plus Kallisto, and you will find stuff coming out every few months or weeks or however long. So I don't know that I have one particular favorite. Although the one in the Learning Center, the Kallisto one, there's one where they look at it, for example, at neuroprogenitor stem cells, and there's a nice walkthrough of that with Zika infected ones that I also use as an example case. But you'll find plenty of examples. And like I said, since questions are coming through to me via email, it might be slow. So I will stay on for a moment until I'm told that it's okay for me to say goodbye. But my information and contact should be up there somewhere, and I'd be able to follow up. So with that said, thanks everybody, and I'll just hang on for a second for questions until they tell me that the stream has been killed.
Thank you very much, Jason. There are still some questions asked, and I'm trying to pass them to you as I can.
Well, actually, now since we're kind of not sharing, I guess you could even just say them, and I'll take them.
Okay. Okay, so one of the questions is surprised to see well connection between Cyverse and R. How the coding of Cyverse appeared in RStudio script?
Sure. So let me see if I can go back and show you a little bit more. I'll share my desktop. So on Cyverse, in the discovery environment, let's go to the home here. If you go to applications, there are a whole bunch of things that we call, it's a system that we call VICE, Visual Interactive Environment. Let me just go to the Learning Center because it will be there. So if you go to learning.cyverse.org and you search for VICE, you will find documentation about this system, which allows you to custom deploy things like Jupyter Lab or RStudio or Shiny apps and things like that, where you provide it a container. And so let me show you how this works. I kind of had the other one running as a backup, but I think it's safe to kill just in case I have to launch it. So let me go to my analyses. I'll kill this. These run, oops, I don't want to go to it. These run for 48 hours. If you need more time, you can ask for more time. But the way that they work is if you search for an application. So for R, we often use Rocker because those are the latest images. And if I wanted to search for Rocker RStudio latest, actually, I want the application, not the analyses. That's okay. There we go. It starts just like any other application where I can go, I select the app, and then I might select any data sets that I want. It's in its own isolated container, but I could start this one with nothing. I could just say next. I could choose, what kind of compute is behind it and say next, and click launch. When I click launch, it's going to come up in my list of analyses, and it's basically spinning up a container where that's running it in an isolated environment. I need to deliberately move data back and forth between those two environments, so it's not necessarily mounting the Cyberse data store, but this is listed as submitting right now. In a few moments, it'll change to running, and then that link, if I click on it now, it's going to tell me that it's still basically booting up. But basically, it's that stuff is booting up, and in a moment, I'll have a custom RStudio interface. I could also customize that container. So like right now, it's like booting up. I have it within a few seconds, right? I could, customize a container so that I have certain R packages already installed or certain scripts already loaded, and it's great for classroom environment. If you're familiar with something, for example, like Binder, where you can do that with like a Jupyter notebook, we can do essentially the same, except that it will not go away after a few minutes. Typically it's 48-hour runtime. It's fully customizable, and you can actually run large resources. So hopefully that answers your question. Other questions?
Oh, yes. I would like to know from where can I access the tutorials or manuals? Can we learn this all by ourselves?
Yep, learning.cyberse.org. Everything I showed you, if you go to learning.cyberse.org, just as I did, you can search for all of those tutorials. And at your own time, there's also videos of me and others going through them, so you'll find that. And we have different online courses. In the fall will be the next large one, but we do usually like a weekly Cyberse webinar, and those are cyclic, but those will be starting up soon. So yeah, you can learn it all on your own.
Brilliant, thank you. And here's another question. Do you have any recent publication that applies the Kallisto and Sleuth workflow in the methodology section?
Yeah, on the tutorial, there are links to papers that I would recommend that go through it. And there are Google groups for it, and I think it's a fairly widely used software. So you can find people who are using it, actively developing it. And on the website for Kallisto and Sleuth, if you Google Kallisto, Sleuth, Rna Seq, the Pca Lab has a really nice website where all those materials and walkthroughs are available too. So you can definitely feel confident trying it on your own and getting these results. And then you can come back to Cyberse if you want to use it on our platform or install the tools yourself wherever you'd like.
Okay, thank you very much, Jason. It looks like we've covered all the questions from the audience. But here's a question from me. I'm an R user, and I know that it is updated at least twice a year. And do you have any knowledge that the most recent R version is compatible with the packages that you showed us?
Yes, I have used it. The one I showed you was like 3.6. I've personally used it on R 4.0 and it was fine. And like I said, they actively develop it, so you should be fine. There may be little code changes, right? But everything should work fine.
Brilliant, thank you. And here's another question from me. So eventually we ended up with the results for 2 replicates for each group. Does it allow us to make any valid conclusions, or?
I mean, it would be interesting, but you have to go back and look at those corrected values. So the Q values. I think you probably have lost some things by only having 2 replicates. I would probably want more data, but it's a little bit fishy that you had those 2 outliers that didn't come in with the rest of them. So I don't know if there was some specific effects there. So it still might be useful. It might point you in the right direction, but I might want more data to really say if I feel confident. Like I would look carefully at the genes that were knocked down to make sure they were knocked down. And does that make sense to me biochemically? Okay, if this is a family of kinases and I see that everything that this regulates is in fact, behaving appropriately, it would at least give me. In this case, I think they might have actually been testing a drug. So maybe it's it's enough to sort of say, I can see some effects that I'm looking for.
Okay, thank you. And how about DNA contamination? We all know that that it might happen because the quality of extraction kits vary from one to another. So how does it affect, from your experience, the results, the outcome of
so in this
RNA seq?
In this case, right, you have your known falciparum transcriptome. So you are only aligning reads that match with significance to the known transcriptome. So contaminants, there may be some things that match by homology accidentally, but I wouldn't be all that concerned about that. You can go look, for example, in things, in some of the outputs to look for reads that didn't map. And then you could ask questions about of the reads that didn't map, because there is a pseudo alignment rate, which we didn't look at to see, how much mapped and how much didn't map. But you could go back and look, and even maybe for unmapped reads, ask the question, is that significantly contamination, or what am I looking at? So those are little details that, for sake of time, we didn't get into, but those are good questions to ask and to make sure you have a sense of how that actually looked, because we went through just the surface treatment of it.
Yes. Thank you. And here's another question. I was wondering if there's any other workflow that you could recommend for us, so we could use and then compare the results obtained with what you just shown us.
So you could go back and use something like Star Aligner, which is also popular. There's also Salmon. There is the Tapetu. So there's a number of things out there. With all of the toolkits, there's never 100% concordance between them. So it's not a bad idea if you have the time to go through and see if another analysis pipeline gives you similar results. You hope that the major important findings are replicable across different types of analyses.
Okay, thank you. And does the workflow that you've just showed us, does it provide sufficient information for making conclusions and going further with the research, or would you rather say that we should also use other workflows to have more diverse, more complete results?
So I think, in terms of completion, right? I don't think this is a fault of the software itself. I wouldn't say that the software is not complete. It takes a specific approach where you're looking at known transcriptomes. But even in the human genome, which like a few weeks ago, they published a complete human genome by Adam Filippi and others, where there are more genes that are identified, and you're going through different regions. So there's always new transcripts that may not be accounted for. So I would say that the less is known about your particular organism, maybe the more paranoid you want to be to sort of say, okay, let me compare different things, let me do different strategies, because there just may be, sort of dark matter that we don't know. Whereas even with human, I remember a paper from a few years ago saying that it's always worth it doing a reference guided alignment and transcriptome, because you will still discover some new things. So it's just a matter of, if you're comfortable about what you're looking for, and you're verifying things, and it's not so much discovery that you're looking for some unknown mechanism, unknown genes, then yes, you might have to cast a wider net. But if you are looking for fairly straightforward things and you're confirming that a pathway is being affected and that your treatment is working or not working, then maybe you can be a bit more conservative and you don't have to uncover every rock. So that might be my personal approach to it. Yeah.
Okay, thank you very much. I'll have a quick look if there is any more question. But no, I don't think so. Thank you very much, Jason, that you found the time for us and gave so much useful information, especially that I'm personally interested in transcriptome analysis because I can see the future in that. And I think that many microbiologists will agree with me. So thank you once again. Your presentation was brilliant. Thank you very much.
Thanks very much, Lucas, and everyone else. Have a great day. Bye.
Thank you. Have a great day too. Thank you. Bye.